Upstate Medical University in Syracuse, New York, invites you to be the informed patient with the podcast that features experts from Central New York's only academic medical center. I'm your host, Amber Smith. A molecular biologist at Upstate received a $3.1 million 5-year grant from the National Institutes of Health to investigate the molecular origins of amyloidosis, which is a group of diseases caused by protein misfolding in the bloodstream. I'm talking about that with Dr. Stewart Loh, a professor of biochemistry and molecular biology at Upstate. Welcome back to The Informed Patient, Dr. Loh.
Thanks, Amber. Great to be here.
I'm anxious to hear about your work, but can you start by describing for us what amyloidosis is and who it affects?
Amyloidosis is actually a family of diseases. It affects people in different ways. It attacks different organs, namely the heart, the kidney, and the liver mostly. And it's caused by proteins that do something called misfold in the bloodstream. So something happens to these proteins in the bloodstream. There's about 15 of them that do this. They form these sort of tangles, aggregates, we call them, which deposit in these kidneys and eventually cause organ failure. Unfortunately, they don't affect the brain. So your cognitive function is okay. It's mainly a heart transplant, liver transplant, kidney transplant are the types of manifestations that one sees.
I've heard about cardiac amyloidosis, but I didn't realize it could also affect the kidney and or liver. In a person, does it typically affect one organ, or might you see all three affected?
You might see all three. Typically, the kidney is actually involved quite a bit with all of these diseases, and that's kind of the basis for our project. That's what sort of fascinated me in the beginning, is that all of them affect the kidney. Some of them affect the heart. That's more serious than the kidney to your health. But there seemed to be some common denominator that involved the kidney with these and other amyloid diseases. So that was our entry into the field.
How do people find out that they have amyloidosis? Are there symptoms?
Yeah, that's really the big question. There are symptoms, but they're often confused with other diseases. These are sort of generic symptoms that you would experience in a lot of different diseases. Maybe swelling, maybe you're having some sort of kidney issues. So what is really needed is a way to diagnose these diseases before they damage the organs to the point where you need to take drastic therapies such as organ transplant. So that's probably, in my opinion, that's the biggest need in how to deal with these diseases, is to identify them early. Because if you do, there are treatments that already exist that will work against some of these diseases, at least. And there's treatments on the horizon that will work. But the detection is really what we're focusing on. And the big question of why. Why does this happen? If we can understand why, then we can also understand things like how to prevent it as well as how to diagnose it.
What is the current thinking about what causes amyloidosis?
It's probably a combination of things. One form of the disease is a kind of a side effect of cancer patients. If one has a certain type of leukemia called multiple myeloma, the cancer cells are producing more of this aberrant form of the protein than you would if you didn't have the disease. So that's one. But another form of the disease, which is called ALECT2, it's caused by aggregation of a protein called LECT2. That one's really mysterious. Often in many diseases, there's a mutation involved. And in this case, everyone who has the disease has a particular mutation, but so does about 40% of the rest of the population. So it plays a role, but we think there's something else. And that's why NIH is funding this type of research, to find what that something else is. That's our hypothesis that's driving this project forward.
So what are you and your collaborators investigating? What is your hypothesis?
Our hypothesis is that there's some other physical force in the body which is causing these proteins to misfold. So if you could shrink yourself down in the blood and look at these proteins as they're floating by, they look like normal proteins when they're in their healthy form. But then their shape changes a little bit, and that exposes a patch that you could call a sticky patch. And if another protein with a similar sticky patch is in the same vicinity, these sticky patches can bind to each other. And then if that keeps happening, then pretty soon you have a long fibril. These things look like twisty pretzel sticks you would buy in a grocery store, except much, much longer. These twisty fibrils, they themselves are quite sticky, so they sort of clump together and then sort of clog up the organ. So the hypothesis is something is initially causing these proteins to deform and expose this sticky patch. It could be things like a mutation. If you're born with a genetic mutation, that makes the protein structurally weaker and allows it to misfold in this way easier. But we think it has something to do with the kidney and the way that the blood flows through the kidney. So when blood or any liquid flows through a tube like a blood vessel, it experiences shear forces which will cause the molecules to a greater or lesser extent to kind of stretch, kind of deform. They kind of elongate as they're traveling through the vessels. So we focused on the kidney because, as I mentioned, this is where a lot of these deposits are found, but also your kidneys filter all your blood. So your blood is just coursing through the kidney, very high flow rates and velocities. So we think this could be sort of the engine that is causing the misfolding of this select group of proteins. So that's our hypothesis.
This is Upstate's The Informed Patient podcast. I'm your host, Amber Smith. I'm talking with Dr. Stewart Loh, a professor of biochemistry and molecular biology at Upstate. So let me understand, do these proteins naturally change their shape in order to fit through the blood vessels?
No, that's a good
Okay.
question. There is no, to my knowledge, physiological role for this. There are in some other proteins, like for clotting proteins, that's part of how they function is they change their shape, and this is part of how they do their job. But to my knowledge, this is not supposed to happen in these proteins, and their role in the body does not require them to undergo this sort of change.
So how will you test your theory?
Well, we've partnered with several investigators at multiple sites to do this. We originally came up with this idea. It was around pandemic time. We couldn't come in to do research, so we were just thinking of new projects to do. And I and a collaborator he was then at Syracuse University. He's now at University of Tennessee. His name is Da-Cheng Ren. He is a engineering professor, a biomolecular engineer. And we came up with the idea of making these kind of artificial kidneys that are very small. They can fit in your hand and they don't look like a kidney, but we put little channels in the kidney that are meant to mimic the blood vessels in the kidney. The blood enters your kidney through a big artery, and then it branches and branches and branches and branches till it finally gets to the glomeruli that do all the filtration. But anyhow, we made these channels branch and branch and branch with the same dimensions. And then we pump our protein through there using a pump that's meant to simulate the pumping action of your heart. And then we sort of see what happens. And what we discovered is that we were able to see these proteins form amyloid fibers, these long sort of pretzel-like sticks. And we were able to see them aggregate and kind of clog up the channels in a way that we were unable to see using conventional ways to get these proteins to aggregate. By conventional, I mean, typically one would take these proteins and put a little stir in there, just sort of beat the heck out of them. You turn the stir up really high. But this is very not physiological. And the types of forces that the protein experiences under those conditions, we think, produce much different structures and much different fibrils and much different types of aggregates, which are not really relevant. So what we're seeing with our device is by flowing it through this kidney-like device, we're seeing different structures. We're seeing effects of mutation and various other kind of physiological insults, if you will. They have effects in our system that we don't see in when we do it by conventional methods of study. So the innovation here is really that microfluidic device.
If I can interrupt, what does this artificial kidney look like? What's it made of?
It's made of a plastic polymer, so they etch these very small channels. They start from about 5 millimeters. You can easily see that with your eye. That's about the diameter of a kidney artery. But then they get very, very small really quickly. It's actually rectangular, and it's mounted on a microscope slide so we can see what's happening in real time with a microscope. And then we put it in a room that's held at body temperature, 37 degrees Celsius. And we pump it and we watch it aggregate under a microscope, and we take pictures and we see how fast and what the types of structures are. But a regular microscope isn't nearly powerful enough for us to see the molecular details. So when the experiment's done, what will happen is as these little mini kidneys get clogged up, the slag will eventually rupture. That sort of mimics a kidney disease in which the kidney gets all congested. But we will remove these fibrils and then look at them under the electron microscope, which is another collaborator which I recruited to look at the molecular details, the atomic structure of this. So that collaborator is from Upstate, who's an electron microscopy specialist. That's Stefan Wilkins. And then we have another collaborator at the University of Texas Southwestern who specializes in determining structures of amyloid fibers from patients with the disease. So the goal is to compare our structures created by our microfluidic device with structures obtained from human beings, which already exist, and see if they're the same. If they are the same, then we will have been the first lab to really be able to get to produce these structures in sufficient quantity and quality to develop diagnostic and therapeutic tools. We have an additional collaborator from the Mayo Clinic in Rochester, Minnesota, named Nelson Leung, and he's a physician who specializes in kidney disease as well as amyloid diseases of the type we're talking about. And his role is to provide our study with samples that were collected from patients that have these diseases. And this is really essential because the studies I've told you about so far are done with purified recombinant protein, not taken from human beings, but synthesized in the laboratory. And it is imperative to cross-check our results with those obtained from clinical human samples.
So you think you might be able to show that it's just the natural force of the proteins in the blood going through these vessels that causes the misfolding?
Yes. That would really be a good test and a proof of our hypothesis. The final proof being, does it generate disease-relevant fibrils? These are the fibrils that cause the disease. The only way to know is to determine their structures at an atomic level. Because if you create fibrils in a test tube by these other methods, and we've done this, and others have done this, and you compare them to the fibrils that you take from patients with the disease, they're often very different. And therefore, any tools that you develop based on the wrong information is not going to give you relevant information for the disease. And my lab also has another major research effort, which we've been doing for many years, which is to develop biosensors, very sensitive molecules that can tell you if a substance is there and how much of it is there. So we're hoping to unify these two projects and develop a blood-based diagnostic assay for identifying these misfolded or incorrectly folded proteins in the blood before they can accumulate to the point where the organ is damaged.
And so that would get back to earlier diagnoses, hopefully,
Mm-hmm.
and earlier treatment.
Yes, the current diagnosis is very long and the answer often comes too late. So if a doctor suspects you have one of these amyloid diseases, they have to go into the organ for a biopsy to take a little piece of that organ out. And then they send it to Mayo Clinic, which is pretty much the only place in the US that will do this. And they will perform a battery of tests. They'll first ask, okay, is there amyloid here? That's not too hard to determine, but that's not good enough. You need to know what type of amyloid is it. Is it protein A, B, C, D, or E? Because the treatments are all different. That type of information is very laborious. And also, by the time you do that assay, the organ is already infiltrated, obviously. So we need to develop something where you can do it in the blood in a more easy test that happens well before it's too late.
Well, your work sounds very encouraging. And it's very interesting, too. Thank you so much for taking time to explain it.
Thank you very much, Amber.
My guest has been Dr. Stewart Loh, a professor of biochemistry and molecular biology at Upstate. The Informed Patient is a podcast covering health, science, and medicine, brought to you by Upstate Medical University in Syracuse, New York, and produced by Jim Howe, with sound engineering by Bill Brokel and graphic design by Dan Cameron. Find our archive of previous episodes at upstate.edu/informed. If you enjoyed this episode, please invite a friend to listen. You can also rate and review the Informed Patient podcast on Apple Podcasts, YouTube, or wherever you tune in. This is your host, Amber Smith, thanking you for listening.